An estimated 31 major pathogens cause 9.4 million episodes of foodborne illness, just shy of 56,000 hospitalizations, and up to 1,351 deaths in the United States each year. 58% of these illnesses are caused by norovirus, followed by non-typhoidal Salmonella, Clostridium perfringens, and Campylobacter. The leading causes of hospitalizations are non-typhoidal Salmonella, norovirus, Campylobacter, and Toxoplasma. The pathogens that cause the most death are non-typhoidal Salmonella, Toxoplasma gondii, Listeria, and norovirus. In 1993, the University of Georgia established the Center for Food Safety with a mission of maintaining and improving the safety of foods through the development of methods that detect, control, or eliminate pathogenic microorganisms or their toxins. The research conducted at the Center for Food Safety helps solve problems in the food industry and provide consumers with high-quality foods with minimal risk to human health. Ensuring a safe and high-quality food supply requires technologies that often influence both food safety and sensory attributes. Developing or changing procedures for processing, preserving, packaging, storing, or distributing foods must also be proven safe. And quality can't be sacrificed in the process. A new technology that increases safety may compromise quality, and a technique that enhances quality may create a greater safety risk. It's a delicate balance. Commercial success of microbiologically safe food products requires an understanding of quality and safety relationships that maintain quality within strict safety constraints. The team of food scientists at the UGA Center for Food Safety has the knowledge and experience to address these issues. Center scientists develop methods to detect and identify foodborne pathogens like Shiga toxin-producing E. coli, Salmonella, and Listeria monocytogenes. Current studies focus on developing intervention methods for controlling foodborne pathogens on produce, poultry, and meats. Other center groups focus on the conditions leading to pre-harvest contamination of produce. In this area, scientists explore the effects of different types of manure and carbon amendments on inactivation of zoonotic pathogens during aerobic composting of animal manures. They identify conditions under which internalization of enteric pathogens into leafy greens can occur if pathogens are present in fields, either via contaminated irrigation water or contaminated soil amendments, and determine the risk of lettuce contamination during field harvest when workers' gloves are exposed to soil and lettuce exudates and when their gloves are periodically washed in sanitizing solutions. The primary pathogens of interest in these studies have been Escherichia coli O157H7 and several strains of Salmonella. A plasmid containing a gene that expresses a green or red fluorescent protein is inserted into these pathogens to facilitate recovery and detection in both laboratory and field studies. Shiga toxin and attachment and effacement virulence genes have been knocked out of several outbreak isolates of E. coli O157H7 in order to use them in field studies. To distinguish between internalized and surface populations of pathogens, both leaves and roots from leafy green plants have been washed in a disinfecting solution of silver nitrate and then ground to a pulp. Fresh fruits and vegetables, as well as some types of nuts, have been implicated as sources of foodborne pathogens in outbreaks of human diseases. Although pecans have not been documented to be involved in these outbreaks, research is being done at the Center for Food Safety to develop and evaluate treatments to eliminate Salmonella and other bacterial pathogens. Of the ground mixture is taken for enumeration possible risks of illnesses associated with
an enrichment
consumption
nutrient
of potentially
medium
contaminated
is added to the
pecans.
mixture to facilitate growth of the
Harvesting,
pathogen,
storage,
for subsequent detection
shelling,
of fluorescent
and processing
cells on an
practices
agar
commonly
plate.
used in the pecan industry are being studied to determine ways to keep nut meats free of microorganisms that could be harmful to consumers. Center research also involves the testing of soil, compost, and processing wastewater for foodborne pathogens on pastured poultry farms. In addition to environmental samples from farms, pasture-raised poultry carcasses are rinsed after processing and tested for two common foodborne pathogens, Campylobacter and Salmonella. Methods used to detect and enumerate these pathogens are those recommended by the United States Department of Agriculture, such as the most probable number method. Human norovirus, commonly referred to as the stomach flu, causes outbreaks of vomiting and diarrhea. A large majority of norovirus illnesses are associated with hospitals, daycares, and nursing home facilities. Noroviruses are the leading cause of foodborne illnesses in the United States, contributing to an estimated 58%, or about 5.5 million illnesses every year, according to the Centers for Disease Control and Prevention. Foodborne illnesses are commonly associated with the consumption of raw oysters, other foods that are consumed raw, such as fresh produce, and prepared foods, also called ready-to-eat foods. Food handlers are often implicated in outbreaks of norovirus. Proper hand washing can greatly minimize the risk of food contamination. Noroviruses can remain infectious on food and food contact surfaces for days to even weeks, and are resistant to hand sanitizers and to many chemicals used to clean surfaces. For this reason, our laboratory focuses on developing and evaluating new sanitizers that can be used on hands, foods, and food contact surfaces. In addition, we are exploring new food processing technologies that can reduce the risk of norovirus contamination of foods. Our laboratory also has a great interest in developing rapid detection technologies that can reduce the time it takes to confirm that a food is safe to eat. Virus culture techniques and molecular biological methods are among the many tools we use to study the transfer to and survival of foodborne viruses after exposure to chemical or physical sanitizing treatments. Another group of foodborne pathogens that can cause illness in humans are the parasites, such as Toxoplasma, Cryptosporidium, Giardia, and Cyclospora. The Center has research projects that include isolation, identification, genotyping, and control of parasites in human and animal populations, the environment, foods, and water. Parasites are hard to kill using common sanitizers and disinfectants, and they are very resistant to various environmental conditions. Because of their complex life cycles, unlike bacteria, they cannot be enriched in laboratory settings, thus making parasite research more challenging. The host response to parasitic infections and development of diagnostic assays is also part of the research done at the Center for Food Safety.